નમસ્કાર, હું આદર્શ છું. વેલકમ ટુ આદર્શ એજ્યુકેશન સોસાયટી. સ્ટેન્ડર્ડ 10th, સબ્જેક્ટ સાયન્સ, ચેપ્ટર 6, life processes. Dear students, today we'll discuss the activity. See the activity here. Take a potted plant with variegated leaves. Variegated means different varieties of colors will be there, green and other combinations, or spotted leaves you are saying. For example, money plant or crotons. And next what you have to do, keep the plant in the dark room for 3 days so that all the starch gets used up. So what will be the whatever the starch is stored, that the plants they are using. Now keep the plant in sunlight for about 6 hours. Okay, then you have to keep under the sunlight for about 6 hours. So then you have to go for plucking. Take out the leaf from the plant, mark the green areas in it and trace them on a sheet of paper. So then you have to take a trace of sheet of paper and mark the green areas. Dip. Next one is you have to dip the leaf in the boiling water for a few minutes. So just you have to go for boiling. After this, immerse in the beaker containing alcohol. Then after the boiling, and you have to dip. Immerse means just dipping, rinsing in the beaker of containing alcohol. Carefully place the above beaker in a water bath or heat till the alcohol begins to boil. So then you have to go for boiling. What happens to the color of the leaf and what is the color of the solution? So ask here what happens to the color of the leaf and what is the color of the solution? Now dip the leaf in the dilute solution of iodine for a few minutes. Take out the leaf and rinse off the iodine solution. Then finally observe the color of the leaf and compare the leaf with the tracing and as well as the real one. And what you can conclude about the presence of the starch. So dear students, what will happen? What happens? See, now we will go for the question wise answers. See, what happens to the color of the leaf? What is the color of the solution? This question. See, what happens to the color of the leaf? What is the color of the solution? The leaf becomes colorless because on boiling the leaf, it loses the green pigment or pigmentation. The water becomes green color due to the presence of chlorophyll that was present in the chlorophyll. Okay. Then observe the leaf, compare with the tracing the leaf done in the beginning. So this one, observe the leaf. The leaf shows blue-black and some areas colorless patches also. So what can you conclude about the presence of the starch? So what we are going to conclusion? The green patch areas become blue-black when they are treated or tested with the iodine. See, you know that when you are adding iodine to the starch, it shows blue-black color. So this is the test for the starch. While the chlorophyll devoid areas, where a chlorophyll is not there, those areas remains colorless. In this way, you can find or you can show that chlorophyll is essential for the photosynthesis. Look here the leaf, which areas becomes blue-black in the colors. Okay. So this one, beginning one, and after the tracing, and then you added the iodine. So this shows these are variegated leaf also. Okay, now next we go through that. Now let us study how the plants obtain carbon dioxide. How the plants are getting the carbon dioxide. In class 9th, you talked about stomata. What is stomata? So what do you mean by stomata? So these are tiny pores present on the surface of the leaves. See, stomata are nothing but They are the tiny pores present on the surface of the leaves. And in this stomata, large amount of gaseous exchange takes place in the leaves through these pores for the purpose of photosynthesis. So you know that plants are what doing the photosynthesis process. In this photosynthesis process, you are knowing they are taking carbon dioxide and oxygen given out. But it's important to note that here that exchange of gases occurs across the surface of the stems, roots, leaves as well as showing that the photosynthesis gaseous exchange not only in the stomata or leaves but also it takes place the plant's roots, stems and leaves as well as. Okay. Since large amounts of water also can also be lost through this stomata. You know that large amount of water also lost through this stomata. So that we are calling transpiration. Okay, the plant closes the pores when it does not. This stomata, the plant closes the stomatal pores when it does not need carbon dioxide for photosynthesis. So you know that in the desert plants, so their temperature is so high. So that is why desert plant closes the stomata during the day and night time they will open and taking the carbon dioxide. So whenever they don't want carbon dioxide, they will close the stomata. The opening and closing of the pore is function of the guard cells. See, look here now. Here the diagram is given. This is the guard cells. This is stomatal pores, and these are closed. So this opening and closing of the stomatal pores are guided by the guard cells. Okay. How are guard cells regulated by the opening and closing? See, here the question arises, how are guard cells regulated by the opening and closing of the stomata? See, for this answer, see, the guard cells, the opening and closing of the pores of the function is of the guard cells. The guard cells swell whenever water flows into them and causing the stomatal pore open. So then these times the stomatal pore is opening, and similarly the pore closes if the guard cells shrink. See here they will close the opening. Similarly, when the pore closes, when they don't want to carbon dioxide, then it will close the stomatal pore. Okay. Now we'll discuss about 6.2 activities. So what is the activity says? Take 2 healthy potted plants. This shows that how will you show that carbon dioxide is necessary for the photosynthesis. Take 2 healthy plants, potted plants, which are really or nearly the same size. And now place each of the plant on a separated glass plates. Place the watch glass containing potassium hydroxide by the side of one of the plants. The potassium hydroxide is used to absorb the carbon dioxide. See the plants here with potassium hydroxide here and without potassium hydroxide. See, nearby this plant, watch glass is containing potassium hydroxide. Here watch glass will be there, and here without the potassium hydroxide. You know that why they are keeping the potassium hydroxide? Because the potassium hydroxide is used to absorb the carbon dioxide. And this bell jars below cover both the plants with separate bell jars as shown in the figure. Use Vaseline to seal the bottom of the jars to the glass plate so that the setup is airtight. See here, you have to tie it with the Vaseline, because what will happen, otherwise air will enter. Keep the plants in the sunlight for about 2 hours. How many hours? 2 hours approximate time. And pluck a leaf from each plant and check for the presence of the starch in the above activity. So you have to go for check with the presence of the starch. The question is there, do both the leaves show the presence of the same amount of starch? So what will say the answer? The leaves of the plant show absence of the starch, while the leaves of the plant show presence of the starch. Do both the leaves the same amount of starch? No. The leaves of the plant shows absence of the starch, while the leaves of the plant the leaves of the plant A shows the absence of starch, while the leaves of the plants B shows the presence of the starch. Okay. What can you conclude from this activity? What you are going to conclude? So starch is not formed in the leaf of the plant kept under the bell jar along with the potassium hydroxide. So here the starch is not formed in the leaf. Why? Because bell jar containing with potassium hydroxide. This is because this potassium hydroxide absorbs carbon dioxide. So they absorb the carbon dioxide from the air. Hence, the plant does not get carbon dioxide, and it cannot undergo photosynthesis. Okay. And from the above activity, what you can conclude? See, what can you conclude from this activity? So we can conclude that a good amount of carbon dioxide is necessary to carry out the photosynthesis at the proper rate. Okay. Now we move on. Based on the two activities performed above, can we design the experiment to demonstrate that sunlight is essential is given. So far, we talked about the how autotrophs and meet their energy requirements. You know that what do you mean by autotrophs and what do you mean by heterotrophs also. So autotrophs means what? The organisms use simple food material obtained from the inorganic sources in the form of carbon dioxide and water. For example, who will come there? For example, green plants and some bacteria will come. And what do you mean by heterotrophs? The organisms utilize complex substances from the other substances and broken down into simple ones. To achieve this, organisms are using biocatalysts, and that is called as enzymes, we are saying. Okay, example will comes animal and fungi. These are the example. So far we have talked about the autotrophs meet their energy requirements, but they also need some other raw materials for building their bodies. See, autotrophs will need even other materials. Okay, water used for the photosynthesis taken up from the soil by the roots in the terrestrial plants. So water from the soil by the roots they are taking, and other materials like nitrogen and phosphorus, iron, magnesium are from the soil. See, these are also required for building the bodies. Which one? Nitrogen, phosphorus, iron and magnesium are also taken up from the soil. See, nitrogen is an essential element used in the synthesis of proteins. See, plants has to get nitrogen is the essential elements for them, proteins and other compounds, for preparing the proteins and other compounds. This is taken up in the form of inorganic nitrates or nitrites. See, you don't know that atmospheric contains nitrogen, but plants can't take the nitrogen. So they have to get the nitrogen in the form of nitrates or nitrites, or it is taken up as inorganic compounds which have been prepared. So this one is prepared by the bacteria. Certain bacteria are converting. So you know that bacteria are converting the nitrogens into nitrates and nitrates from the rhizopus bacteria you are knowing. Okay, by symbiosis bacteria from the atmospheric nitrogen. So then only the plants get essential nitrogen. Okay, dear students. Now we'll discuss remaining in the next video. Okay, until that, bye bye. Thank you for joining us.